morning to everyone. The title of my lecture is Troubleshooting in Spawn and Mushroom Production. This is the outline of my presentation. Introduction, review of microbiological techniques, and problems in mushroom and spawn production, and recommended solution. At the end of my lecture, we're expected to be able to enumerate and explain the different problems that can be encountered in spawn and mushroom production and discuss how the problems can be prevented. Similar to other crop production, mushroom production is not always a better process. Several problems may be encountered during cultivation phases such as microbial contamination by bacteria and fungi, insect infestation, and abnormal growth of the fruiting bodies. In the previous lectures, you have learned some microbiological techniques which are essential in mushroom production such as sterilization, pasteurization, and aseptic technique. Sterilization can be defined as the process of killing all forms of life. In media preparation, the different ingredients such as potato, sugar, and agar contain microorganisms. These microorganisms can be destroyed by sterilization. In the laboratory, we use autoclave or sterilizer for this purpose. The sterilization conditions are as follows: pressure of 15 pounds per square inch, temperature of 121 degrees Celsius for a period of 30 minutes. Pasteurization, on the other hand, is the partial sterilization of substrate. In the CLSU Mushroom Center, we use fabricated pasteurizer or drum for this purpose. The temperature is 60 to 80 degrees Celsius for a period of 8 hours. Another technique is aseptic technique. Aseptic technique is the use of practices and procedures to prevent contamination. This is usually performed in inoculation of culture media and grain spawn and tissue culture of mushroom organism. When doing tissue culture and inoculation activities, always disinfect the working area with 70% ethyl alcohol or 70% isopropyl alcohol. This activity should be done inside the laminar flow or inoculation chamber. Like other crops, mushroom may be contaminated by fungi and bacteria and attacked by pests such as insect pests, so that proper sanitation and hygienic practice should be strictly observed. Also, abiotic factors such as temperature, moisture, light, and ventilation may affect growth and development of mushrooms. Bacterial contamination. Bacterial contaminants grow on the culture media, usually a day after inoculation. Bacterial growth can be characterized to be slimy and water-soaked growth, especially if the slanted medium has excess water. There are two sources of bacterial contamination. If 100% is contaminated, the cause is under sterilization. However, if 10 to 50% is contaminated, the problem is due to the technician. He has to improve his skills and practice proper asepsis. The second problem is fungal contamination inside the bag. There are two causes of fungal contamination inside the bag. When the contaminant originated from the opening, the problem is due to contaminated grain spawn. However, if the contaminants are located in the entire bag, the problem is due to under-pasteurization. Third is fungal contamination in cotton. Wet cotton results from abrupt opening of the pasteurizer or autoclave. During incubation, this plug gets contaminated with airborne fungi such as Neurospora. To prevent this problem, cover the cotton plug with a clean paper and avoid immediate opening of the cover of the pasteurizer. Next is bacterial contamination of the bag. This problem is due to bacterial contamination. This can be recognized when the bag is almost fully ramified by mycelia of the mushroom. As you can see, a demarcation line can be observed between the mycelia and the colonized substrate. And this happens when the moisture content is higher than 65%. Another problem is trichoderma contamination of the fruiting bag. The growth of trichoderma is characterized by green powdery growth with white irregular margin. This fungus is usually airborne and present when the condition is humid with insufficient ventilation. The next is contamination by slime molds. Slime molds become apparent on a spent fruiting bag which has been left unattended inside the growing house. These molds may be dispersed by water droplets and they grow when there is excess water in the bag. Next is bacterial contamination of the grain spawn. This problem is due to high moisture content. Next is insect infestation. Flies are a common problem in mushroom houses where improper sanitation is not being practiced, especially during harvesting. Avoid leaving parts of the harvested mushroom from the fruiting bag. The remnants become inviting to the attack of these flies. Make sure that the screen of the house is properly installed to prevent introduction of this pest. The next problem is elongated stipe and delayed expansion of the peleus. The abnormal elongation of the stipe coupled with non-expansion of the peleus may be attributed to insufficient ventilation and light inside the growing house. Thank you very much for your attention.